Alrighty. Getting started here, right where we left off with chemical synapses, and we're going to start today with neurotransmitters. So there will be some topics from here on in this chapter and in chapter 12 that we're going to omit just for time's sake. I tend to not omit things because I think it causes confusion on your behalf. Like, you're not sure, like, do I know this, should I not know it? And usually everything is fair game. But for time's sake in lecture and just for overall comprehension, there are going to be a few things that will cross off. So what I'll do is I will go through and delete the slides from the PowerPoint that I tell you to cross off, and I'll upload like a kind of a key PowerPoint, one that has all the things on it that are already taken off. That way you know which ones are which. So yeah, so I'll do that kind of as we go. Go ahead.
So if we cross them off where you don't need to know it?
Correct.
Okay.
Correct. Yeah. So and again, it causes, the reason I don't like to do it is because it causes confusion when you look back in your book, right? Because you might be reading your book or interacting with your book, and you're like, oh wait, I just read this whole section. I didn't have to know it because I crossed it off, right? So I apologize for that, but it's meant to save you time in the long run. So just kind of make notes of the things that we cross off in here. So the neurotransmitters, we've talked about acetylcholine already, so we're kind of familiar with the idea of neurotransmitters. These are the chemical signals or chemical messengers that are released at a chemical synapse. And so they're not all the same. There are different types of neurotransmitters. And so there's different types of neurotransmitter receptors. And each cell can have receptors for different neurotransmitters. In other words, each postsynaptic cell can receive some of these and some of these, right? It can receive different neurotransmitters, and then it can respond accordingly, right? It can respond depending on which neurotransmitters it was sent. So they're released from the axon terminals, they cross into the synapse, and ultimately they bind to specific receptors. So if we recall, we had our, and I'm just redrawing this for our purposes today, but you probably have this somewhere in your notes, right? This is our presynaptic cell and our postsynaptic cell. And so on this postsynaptic cell, there will be specialized receptors for various neurotransmitters. And so this presynaptic cell might release neurotransmitter A, neurotransmitter B, neurotransmitter C, right? It could release all of the above, right? And so this postsynaptic cell has to interpret the signals that it's being sent, right? And those signals are either excitatory or inhibitory, right? They're either going to turn on the cell or turn off the cell. So we are crossing this one off, ionotropic receptors and metaboretropic receptors. You don't have to know the difference in those receptors, so we're skipping over that. We're also going to skip over this one, which explains in a little more detail about the metabotropic receptors. You don't have to know the difference between them. I'm going to tell you everything you need to know about the receptors as we go. So we're crossing, oh my goodness, crossing all of these off. Okay, so we get to this section about major neurotransmitters. So neurotransmitters, again, can cause an excitatory postsynaptic potential or an inhibitory postsynaptic potential, depending on which one, right? And that's how that signal is going to be sent. That the action potential comes down, right? And it could trigger the release of an excitatory neurotransmitter or an inhibitory neurotransmitter. And that's going to cause either a depolarization or a hyperpolarization on our postsynaptic cell. So the neurotransmitter that binds is going to dictate what this does. Is it going to send a signal, or is it going to hyperpolarize so it makes sure that it doesn't send a signal, right? So our neurotransmitters are classified into some different categories based on their chemical structures. You don't have to know the different chemical structures. You only have to know certain neurotransmitters, and we'll go through them. So this chart comes from your book. It lists all the neurotransmitters. You don't have to know them all. I'm not going to say you need to know this. There will be times when I just point to a chart and say you need to know that. This is not that time. You do need to understand the different types of synapses that exist in our nervous system, though. So we have, so these are chemical synapses that we're talking about, right? Chemical synapses use a neurotransmitter because our electrical synapses just use a gap junction, right? Those sodium and potassium ions just flow right through a electrical synapse. But a chemical synapse could be a cholinergic synapse. A cholinergic synapse is one that uses acetylcholine. Right? It has ACHE to recycle ACH. Right? Usually, this is an excitatory synapse. A synapse that uses acetylcholine is excitatory. So in my weird little Jamie brain, whenever I think of excitatory things, I think of screaming them or yelling them or getting excited saying them. And so the way you do that on paper is you add an exclamation point. So I always think of cholinergic, right? And if you say it with lots of energy, it reminds you that it's excitatory, right? So be excited when you say cholinergic, or when you write it, you can be excited with just adding an exclamation point. And that might help you remember that that's what it is, right? So we also have our other types of chemical synapses. I don't think it even says it on this slide, but they're called adrenergic receptors. Adrenergic receptors use epinephrine and norepinephrine. So these are using epinephrine and norepinephrine as opposed to acetylcholine. And so these could be excitatory or inhibitory. So these could be either excitatory or inhibitory. So most of our synapses are going to be classified as either cholinergic, excitatory using acetylcholine, or adrenergic using epinephrine and norepinephrine, and they can either inhibit or excite depending on where they are in the body. And we'll look at these adrenergic receptors more when we do the autonomic nervous system towards the end of the semester. So these are just other neurotransmitters. We're skipping over them, so you can cross this one off. The neurotransmitters that you should know are the ones up here: ACH, epinephrine, norepinephrine. Right. So we'll learn about some of these as we go through and look at some different processes, but learning them in isolation and memorizing what they are isn't really important for our level. So of course, all of these neurotransmitters are kept in balance in our body, just like everything else, right? Homeostasis, homeostatic mechanisms. But when these neurotransmitters are created out of imbalance, right, or they're overproduced or underproduced, that's when psychological disorders could occur, right? So there's a whole range of medicine dedicated to psychoanalysis and understanding how these neurotransmitters affect how you feel and your personality and the things that you experience in your mind, in your head. And so psychiatric disorders like schizophrenia and depressive disorders are all linked to neurotransmitter imbalances. You don't have to know which neurotransmitter, I'm just sharing that for general knowledge. Same thing, anxiety and bipolar also linked to this. If these were given on a case study, for example, I don't want you to feel like you have to know all of this information. I would give you information, and then you'd have to interpret it. So I might say, for example, and this is not the case study because I haven't even made your exam yet, but let's just say, Jamie has an anxiety disorder, right? And this anxiety disorder is because of a GABA activity, right? There's a imbalance in the GABA neurotransmitter activity, right? Question 1, what is a neurotransmitter? Question 2, what type of synapse are we talking about here, right? Question 3, describe the depolarization of the blah, 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 right? I would ask you questions like that. So I'm not going to ask you specifically about these things. They're just in there to say, oh, that's what it is, right, for everyday knowledge. So all of our neurons, this is kind of the last little piece of understanding general neuroanatomy, is the idea that our neurons can work together in pools. It's kind of like, You know when you used to do like group work in high school, and like one of you would be the timekeeper, and one of you would be the recorder, and one of you would be kind of the presenter, right? You would all have different roles. Well, it's kind of like that. Neuronal pools are different groups of neurons working together to do the same task, right? And so these neuronal pools can be created differently. There's different formations of them for different purposes, right? Because there's always a structure and function relationship in the body. So they're basically complex connections between multiple neurons. And so we'll look at some of these neuronal pools and how they're formed. I'm actually going to erase this guy so we can talk about these neuronal pools and how they're put together. So almost everything in our body is controlled by a neuronal pool, a pool of neurons. It's not just one single neuron that's regulating or monitoring something. Almost all of it is using multiple neurons. So here is an example of a neuronal pool where you have one neuron communicating with multiple neurons, right? So this is kind of like, mass communication here. And then these neurons would go out and spread the word, right? Whatever it is, contract the bicep muscle or whatever. So this is what we're talking about when we're talking about a neuronal pool, a group of neurons working together. So one is called a diverging circuit. And that's what we just saw. A diverging circuit is when you have a single neuron. This is my single neuron. Kind of looks like a dude with no arms. Right, a single neuron branching into multiple. Right, a single neuron branching into multiple. So this would allow a single neuron to communicate with multiple parts of the body. This would be good in mass communication. Because think about it, if the signal coming into your brain says it's cold, right? You're not going to get goosebumps on one part of your body, right? You're going to get goosebumps everywhere on your body. So that's divergent, right? The signal from your brain saying we need to get goosebumps is going to go down and out and spread to all of the neurons connected to all of those erector pili muscles. So that's divergent, right? One going to many. Most of the circuits coming out of the brain are divergent. Okay, convergent is the opposite. Right, so most of this divergent is from the brain to the periphery. Here, we have most of the information going from the periphery to the brain, right? So everything's converging. So here we have multiple neurons, and the information this time is flowing up into one important neuron, right? The information's flowing up and converging onto one neuron. So we used a word a while ago when I was just introducing you to the idea of information coming into the brain and information leaving the brain. Information going from the brain to the periphery is what we call efferent information. Efferent. I'm just introducing the word to you again because you're going to see it more and more. And this we call afferent information.
Can I ask a question?
Yeah, go for it.
Urgent one. So is the information going from the dendrites of those?
No, I drew them backwards. Sorry. Good point. I was just showing that there are multiple, but yes, thank you. So it would be like this, an upside down dude. Yeah, they never travel in that direction. I should have paid more attention to how I was drawing that. Yeah, the information never flows from the axon up to the dendrites. It always flows from the dendrites down the axon, right? Always down the axon. So that's our example here. Afferent information. Goodness. All right. So our circuits can be inhibitory. Inhibitory would be providing negative feedback about something, turning something off, right? Turning the signal off, right? That's what negative feedback does, turns the signal off. And in some cases, synaptic fatigue will cause this inhibition. So synaptic fatigue is when that signal transmission, the communication at the synapse just becomes weaker and weaker and weaker because there's been so much overload. This is kind of like when you first cut your finger, right? It hurts really, really bad, but then after an hour or so, you kind of forget that it's hurting. It's because those pain receptors just kind of say, all right, enough. We know it hurts. Yeah, move on, right? It's that kind of building a tolerance to some signal coming in. And so your synapses can do that if they're exposed to something for a long period of time. Epilepsy is when the electrical activity in a neuron is uncontrolled and can result in sudden bursts of electrical activity instead of this nice organized pattern of circuitry that your body has arranged. Several different neurons can fire at the same time, which results in seizures, right? That's what seizures are. So that excitation overwhelms the circuitry and you end up with the seizure. Right? And so there's different types of seizures. You don't have to know the different types of seizures. The only thing that you should understand is that seizures result when these circuits are uncontrolled, right? When there's random excitatory signals being sent, causing lots of excitation in the neurons. So there's lots of stimulation with an epileptic seizure. So we're going to move into our next chapter here, which is chapter 12. At first, I told some of you guys, I was like, oh yeah, we can probably end early tonight because it's a short lecture, but I looked ahead and chapter 12 is kind of a bear. We're going to end up doing some of it in lab. Obviously not next week after the practical, but the following week we'll end up doing some of it in lab just to make up some lecture time because we're just going to have to. So we're now moving into the central nervous system. So your book kind of divides the nervous system into all of the nuts and bolts, which is what we just finished, right? We understand the structure of a neuron, we understand how neurons work, we understand how neurons work together, right, and communicate together. Now we're going to look at the whole system. We're going to look at the brain and spinal cord together, and then in the next chapter we'll look at all the peripheral stuff. And then in the chapter after that, we put it all together, right? Remember I said we're pretty much in the nervous system for the rest of the semester. So this is the next piece, the central nervous system. And by central nervous system, we're talking about the brain and the spinal cord, right? So those are the two key players here. Of course, this is all dealing with homeostasis, regulation on the major level. This is also your mind, right? This is the inner consciousness, right? Inner control. So we're going to look at how all of that works. So overall, functionally, our central nervous system can be broken down into motor and sensory. And we can see that here, right? If we're talking about all of this divergent stuff going from the brain out into the periphery, would that be sensory or motor? Going from the brain to the periphery? Motor, right? Motor commands coming from the brain, going out to the skeletal muscles. And likewise, all of these convergent circuits, right, bringing information from the periphery to the brain, right? This would be our sensory information, all of our sensory information converging, all of our motor commands diverging. So in this chapter, we're going to see how all of that works. But before we can do that, we have to understand the brain and the parts of the brain and where all of that information comes from. So ultimately, our brain is the integrative center of our body. It is controlling and taking in all the things. Your brain right now knows exactly what your blood pH is, what your blood pressure is, how much carbon dioxide is in your blood, how much glucose is in your blood, how much food is in your stomach, the temperature of the room, whether or not you made out with your boyfriend this morning, right? Your brain knows all of those things, right? And it's processing it, and your brain has to figure out what is important to think about right now, right? My voice, right? What is important to think about right now and what can be ignored, right? Maybe the fact that you have a wedgie right now can be ignored, but maybe the fact that I'm telling you this important stuff can't be ignored, right? Your brain has to figure that out. Interpreting, planning, monitoring, all the things. So let's start with the brain. All right. In general, this is it's continuous with the spinal cord, so you'll see this a little bit in lab. If you go to the human dissection, you'll see the brain there, and you'll see how it is connected to the spinal cord. It's a pretty cool thing. Happens right at the foramen magnum, right? That's that transition. So our brain actually has internal cavities and openings called ventricles. So these ventricles are actually filled with fluid. So the majority of your brain is water, right? And that water, that liquid component helps to cushion your brain, and we'll look at all of that, how it works. Your brain at any given time is getting 20% of your total blood volume, right? Guess how much blood volume your brain gets when you're exercising, when you're breathing really hard and you're huffing and puffing. How much percent does your brain get? 20%. When you're sleeping and you're not moving at all and you're laying there, how much blood does your brain get? 20%. You have mechanisms in your cardiovascular system that make sure, no matter what, your brain is getting 20% of your blood volume at any given time. Why? Because 20% blood volume means the perfect amount of blood glucose and the perfect amount of oxygen for your brain to do all the things that it needs to do. Your brain needs two things in order to survive and function: oxygen and glucose. And without either of those, it's not going to work right. You know that if you've ever been really, really hungry, right? Sorry, I can't think straight, my brain. I'm so hungry, I haven't eaten it, since this morning, right? You know that. Your blood glucose drops, you have trouble focusing. You also know that if your brain has been deprived of oxygen, maybe you went into cardiac arrest, right? Something like that, restricting blood flow to the brain, you could wake up and be brain dead, right? There's no activity in the brain after that. So we're going to go through and talk about each part of the brain. There are 4 major divisions. We're going to graph it out over there. We'll talk about each of them. Make sure you understand them all. So this is going to get pretty big. If you like to draw along with me, make sure you have a full page. And you might even want to turn it landscape, because that helps. So we'll start with the cerebrum. The cerebrum is the brainy part of the brain. When I said we're going to talk about the brain, this is probably what you pictured, right? It's that kind of nook and cranny, wrinkly part of the brain that you're thinking about. So it's called the cerebrum, and it's divided into hemispheres. Right, the right and left hemispheres. This is where all of our higher level functioning is. This is where all the thinking goes on. This is your conscious awareness, right? You hear my voice because there's an area of your cerebral cortex, of your cerebrum, that says, oh, this is what Jamie's saying, right? You see me because there's an area on your cerebral cortex that says, oh, I see light. I recognize that as a word, right? Your cerebral cortex is all of the consciousness. This is what makes you, you, right? Your personality, all of those things comes from this area. Language, right? Recognition, memory, all of those things right here in the cerebrum. So I'm just going to write you and hope you know what that means. The diencephalon, this is the part you've probably never heard of before, but it's actually the coolest part of the brain. And yes, I'm only saying that because I'm a super nerd. Diencephalon. Diencephalon. This is where all of your homeostasis stuff happens. This is where a lot of the regulation occurs. Your sleep-wake cycles, right? Your hormones. All of those kind of regulatory things come from this area. So it's regulation, and I call it a relay center because your diencephalon gets a lot of information, and your diencephalon will say, okay, the cerebrum doesn't really need to know that right now because the cerebrum's busy doing something else, right? So it relays information to the cerebrum when appropriate, right? So it's not telling the cerebrum things all the time. Whoa. The cerebellum our cerebellum, this is also divided into right and left hemispheres. This is our balance and coordination. Animals that, they're called arboreal animals. Animals that live in a tree, like squirrels, they have really large cerebellums. They have very small cerebrums, but they have very large cerebellums because they have to balance on those little twigs, right? Cats have a really big cerebellum. We'll have a chance to dissect a brain in lab, and the brain that we have is, I think it's a sheep brain, and this sheep brain has a, it's small compared to ours, but the ratio of cerebellum to cerebrum is about the same on a sheep as it is in us. And then we have the brainstem. The brainstem is all of the life functions. So the things that make you alive. Your heart rate, your respiration rate, all that stuff is managed right there in the brainstem. And your brainstem is also the last stop on the bus out of your brain, right? So this is the very bottom of your brain. This is the connection between all of the stuff up top and your spinal cord. So everything has to pass through here. Everything that's going to work its way up has to pass through the brainstem. So there are some of the things. This is a summary of what we just said. We're going to get a little more in depth now. Talk a minute about the spinal cord. So our spinal cord connects directly to the brainstem, and it actually ends right at our lumbar vertebrae. So most people think that the spinal cord, goes all the way down, but really the spinal cord ends at your lumbar vertebrae, and then it just kind of frays out, right? Almost like a string that hasn't been sealed on the end. That's how your spinal cord looks. So it's about 18 inches long, only about 1.5 feet long, and it has in it a canal, a central canal. So there's actually an opening in the middle of your spinal cord for cerebral spinal fluid to pass. This is our spinal cord. Our spinal cord has, and we did this drawing briefly before, just so that I could talk to you about gray matter and white matter, but we're doing it again. So if you recall, white matter are myelinated axons, gray matter is cell bodies. Our spinal cord, this little area right there is our central canal. It's where cerebral spinal fluid circulates. White matter is on the outside of our spinal cord. This is all of our myelinated axons. So this, I think of this as like a superhighway. Stuff's coming and going, up and down. This is a cross-section of your spinal cord, as if you were to cut in a transverse section and look into someone's spinal cord. This is what you would see. Right, we're looking into the spinal cord here. These are all axons going up to the brain, coming down from the brain, all over the place. And so they're arranged, does it tell us here? Yeah. So they're arranged in tracks. So tracks are like bundles, right? And so there'll be like I'll use a different color. There'll be a bundle of nerves here that go to a specific area of the brain, and a bundle of nerves there that go to another area of the brain, and another bundle of nerves there that maybe come down from one region of the brain. So it is highly organized, right? It's not just like a tangled up network of strings, right? It's very organized in how it's put together. But the cell bodies are here in the gray matter. The cell bodies are there. All of the myelinated axons are here. So it's kind of like the cell bodies are here, and then they kind of swoop up, and then they go up to where they need to go whoop, and they swoop down to where they need to be. So the cell bodies are in here, and then the axons extend into the white matter. And the white matter can either be ascending and go up to the brain, or it can be descending and go down and out to the body. Oops. Now remember, in the central nervous system, we call groups of cell bodies, we call them nuclei. In the periphery, we call them ganglia, right? You have a ganglia right here by your medial epicondyle. But in your central nervous system, we call them nuclei. So we say the nuclei are in here, right? We're saying nuclei meaning a cluster of cell bodies. It's not the same way we call a nucleus of a cell, right? A nucleus of a cell is not the same thing as a nuclei. So gray matter, this is where all of those nuclei are. And so our spinal cord is in the center. The gray matter is in the center. The white matter surrounds it. On our brain, this is a cross section of our brain. Let's see how good I do. Not too bad. So if this is a cross section of the brain, it's going to be put together in the opposite way, where we have gray matter here and white matter on the inside. And that's again because the cell bodies would be up here and they will be projecting down, right? The information comes from the top and goes down. And so the gray matter is where our cell bodies are. It's gray because they're unmyelinated. The whiteness is because of the myelinated axons. So this is a cross section. This is what we drew, right? But the idea here is that it's reversed, right? Gray on the outside, white on the inside, white on the inside, or white on the outside, gray on the inside. And notice the central canal. Notice the holes, our ventricles of our brain. We get big ventricles here, big ventricles here. Lots of holes in your brain. Holds cerebral spinal fluid. All right. Interestingly enough, all of our brains are different. Male and female brains are different. It's because of a process called neuroplasticity. Neuroplasticity basically tells us that our brains are already hardwired for certain things. So, for instance, don't hate me guys, but women tend to be better at multitasking. They tend to be better at handling multiple things. I can cook dinner, I can check homework, I can be listening to, talking on the phone, right? I can do all of these things at once. But what my husband does is he cooks dinner. And the kids tear apart the house. and homework doesn't get done because he's cooking dinner. Right? One task at a time. I'm not saying that to make fun of him. I'm saying it to show you this concept of neuroplasticity. And I bet you'll see other examples of this throughout your life. Guys tend to be better at certain things. Girls tend to be better at certain things, right? And it has to do with this neural circuitry and how we're actually hardwired. Is some of that hardwiring created from our upbringing and we're kind of taught to be like girls or taught to be like boys? That's kind of up for debate. Right? There's still this kind of nature and nurture issue when it comes to gender. But there does seem to be some weight to it, some differences between male brains and female brains. So just wanted to bring that up to you. All right, let's talk about how the brain develops. So normally I show a video, but due to time constraints, we're going to do some imagination. So your brain grows in utero like a flower sprouting from a seed. So imagine a little seed buried in the dirt, right? This seed eventually sprouts a little green thing that grows up through the soil, and eventually you get some leaves, and then some more leaves, and then a flower kind of blooms and blossoms at the top, right? That's exactly how your brain grows in development. The circuitry for your brain, the neural tube, is laid down at, say up here, yeah, I was going to say about 4 weeks. 4 weeks gestation. So if you've ever been pregnant, or you know someone who's been pregnant, they don't know they're pregnant at 4 weeks gestation because they haven't even missed a period yet. And missed periods is how you know you're pregnant, unless you're trying and you're testing and blah, 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 right? But at 4 weeks, the neuroseal has already been laid down. The group of cells that's going to be the brain, the seed of the brain, if you will, is already there, right? And it blooms and blossoms very, very quickly because that neural tube is going to lay the foundation for everything else, right? Which is why taking prenatal vitamins, specifically folate, folic acid, is super important. Folic acid is the vitamin that fuels this development, right? So as it's blooming, it's going to kind of blossom in this pattern that we call vesicles. Vesicles are bubbles, right? We've talked about vesicles that contain ACH, right? Vesicles are bubbles, and that's exactly what happens. This seed elongates, and then little bubbles form. And those bubbles are the different parts of the brain. So there's 4 major bubbles that grow off of this tube, and those bubbles continue to grow and enlarge and wrap around each other until wham bam, there's a brain, right? And so the entire brain is done forming right around the end of the first trimester. The brain completely done. It's one of the first things that's completely done, right? No bones yet, definitely no muscles yet, but you have a brain, a fully functional brain. Super cool how it works. So there are, this is, you don't have to know all of these words, but it's really, really cool. Developmental biology was the thing that I got my master's in, so this is like one of my favorite things, and we're skipping over it, but it's really cool if you want to look it up. So yeah, we're skipping over all the names of the vesicles, right? The idea of a neural tube, right? You don't have to know all that. This shows you the progression, but you can kind of see what I'm talking about here. This kind of blooming and blossoming, and then bam, all brain. Really cool. It's all regulated by these genes. It's super duper cool. All right. One day this board won't freak out on me. All right, let's talk about the cerebrum in detail. So the cerebrum has on it something called sulci and something called gyri. The sulci are the grooves. The sulci are the ditches. So the sulcus is singular, sulci is plural, right? It's that Latin ending. So the sulcus is a single groove on the brain. So if you imagine squishing that brain together, those little grooves, those indentations are called sulci. The mounds are called gyri, G-Y-R-U-S, gyrus, singular, gyri, plural, right? The ending just changes, but the root stays the same. I remember this because I sulk in the sulci, right? When you sulk, you're like low, and I jump over the gyri, right? Whatever helps. I think of jumping over the gyri because you're going to jump over that mound and sulk in the sulci. All right. There's also fissures on our cerebrum. So fissures are really deep grooves. So the gyri are just the everyday little wrinkles in there, but the fissures are the deep, deep crevices. So you have one major one called the longitudinal fissure. The longitudinal fissure is what divides your right and left hemispheres. So the longitudinal fissure is that separation between right and left halves of the brain, right? The deepest groove. So here you see some of the features, and we'll talk about the different lobes and everything, and we'll add to our chart. We just barely got it started. There's that longitudinal fissure down the middle, left and right hemispheres. You see lots of gyri in there, and some of the sulci. So some of the gyri have specific names, and you'll need to know them, and eventually in lab you'll recognize them on models and so on and so forth. And same thing with the sulci. Some of them have specific names, and you'll know them eventually also. But each hemisphere of our brain has these lobes. So we have a frontal lobe, a parietal lobe. What do you notice about these lobes? Same name as bones. Yeah, same name as our bones, right? Yay! Occipital. So we didn't have a bone called the insula because the insula is actually an inside, an interior lobe. So there they are. And they're in the exact locations you would predict them to be based on the bones, right? Frontal bones here, or frontal lobes here, parietal lobes here, temporal lobe, occipital lobe. So the frontal lobes, this is our planning, our logic, our execution, right? This is a lot of your personality in that frontal lobe. This is your consciousness. This is your personality. Planning, personality, all of those things. Consciousness, your awareness, where you're directing your attention, all right here from the frontal lobe. The parietal lobes This is a lot of integrating. So this is a lot of processing and integrating of sensory information. So I'm going to add the word somatic sensory because this is the sensory information that we're consciously aware of. So you're consciously aware of where your arm is right now. You're consciously aware of where your feet are right now because of your parietal lobes. Your parietal lobes give that information to your awareness, makes you aware. The temporal lobes, this is hearing and language, which makes sense because hearing and language go together, right? If you can't hear, it's very hard for you to speak. It's very hard for you to produce and replicate sounds. Right? You know this if you've ever talked to someone who's deaf. One of my neighbors, former neighbors because we just moved, he was deaf and dumb, so he didn't speak or hear, and he was from another country, and he did not speak American Sign Language. So when we would talk with him, it was annoying at first because it was a lot to try to communicate simple sentences. It was like playing charades. He would come up to me and he would do this, and that meant, "Where's your husband?" Right? He's pointing to his wedding ring. So little things like you had to kind of decode, and then eventually we figured out, we could just text each other, right? Because he could speak English, but he couldn't read lips because he didn't know what the words were, but he could write it. So then we would be like this. Okay, yeah, I'll be right back. It was a real cool learning experience, to say the least. So language, and because it's language, this is also memory and emotions. And this is key, I think. Look, memory and emotions are right next to each other in your brain. Did you know that the more heightened your emotions are, the better your memory is? Think about what you did like 3 Wednesdays ago. I don't know, right? But think about the last time you had a really bad day and you were really pissed off, or a day that you ugly cried because something awful happened. You remember it. You might even remember what you wore. You might remember what you were eating. You might remember where you were sitting when you had that ugly cry. It's because your emotions and your memory are linked. So get really, really pissed at me and study. I want you all to hate me and then study your butts off. Okay, occipital lobes, this is vision and everything dealing with vision. So this is what we call our visual cortex. And then the insula, this is our taste, our sense of taste. And this is a lot of our internal organs, the viscera. So you'll feel the fact that your stomach is full. You'll feel that bloated feeling. You'll feel cramps, right? You'll feel those things in your insula. And your insula is where all of that goes. All right, so there's a picture of where they are. So the gray matter on the cerebral cortex, again, this is all that outside piece, right? There's neurons there, but there's also interneurons there. Or I'm sorry, not interneurons, neuroglia there. Remember neuroglia? The astrocytes, the ependymal cells, the microglial cells. All of those cells are also mixed in here with our neurons because our neurons need to be nourished and protected. And so there's lots of other layers to it other than just a bunch of neurons stuck together. All of the neurons in our cerebral cortex are what we call interneurons. What is an interneuron? They're all interneurons. I'll give you a hint. Yeah, I guess I can erase this. Afferent information. I'm using a word that we haven't really learned yet because I want you to practice it. What type of information is afferent? Sensory, good. So this is sensory information. Sensory information comes in to the brain. And then efferent information leaves the brain. Our brain is where all the interneurons are. Interneurons are the decision makers. Interneurons are the interpreters. Interneurons are the ones that are going to decide, are you going to respond to that sensory information, or are you going to ignore that sensory information? Interneurons do that processing. So interneurons are kind of like little cellular brains, right? They're cellular brains. They take the information that they have, and then they decide what type of response, if any, right? Sensory, motor. Sensory information comes in, your brain decides what to do. Your brain made of interneurons decides what to do. Motor commands follow. So, we have a few special areas of our brain. So we understand the lobes, but then there's these things called cortices or cortexes, right? Cortices is plural for cortex. So we have something called the primary motor cortex. Find the blue. Primary motor cortex. Guess what this is? Cortices. Primary motor. This is where all of your motor commands initiate. Your writing right now is all coming from your primary motor cortex. It's a motor command, right? Turning your head, motor command, all coming from your primary motor cortex. The primary motor cortex is on your frontal lobe. You have another thing called the primary sensory cortex. This is all of your somatic sensory information. All of the sensory information that you're aware of comes from here. Or goes there, I should say. All somatic sensory goes here. If you're consciously aware of it, that's where it's hitting your brain. This is on your parietal lobe. And then we have what we call association areas. And we have a few of those spread throughout. We have different association areas that are associated with different special senses. So you have an association area for speech. You have an association area for language. You have an association area for vision. You have an association area for audio. When you think of the word association area, I think of linking it to what it is. I see this thing in my hand. It's white and blue, and it writes stuff. I associate that with a marker, right? It's your recognition of what it is, right? You see a letter that looks like this, a line and a curve, and you associate it with the letter D. And then another area of your brain associates the letter D with the sound D, right? So association is kind of linking the concepts together to give meaning to something, right? It's one of those things that can kind of blow your mind if you stop to think about it. But at one point in your life, you didn't know what things were. You were walking around life not saying, that's a tree, that's grass, that's a dog, that's a curve, this is a sidewalk, I'm late for class. You didn't have that internal monologue going on in your brain. What the heck were you thinking? What do you think when you're 2 and you don't have language for things? What do you think when you're 1 and you don't have language for things? That's like the $1 million question. I mean, people research that all day long. It's really cool to think about. They don't have words for things, and they don't have the association areas for those things. Learning, the process of touching a hot stove and learning that it's hot and associating stoves with hotness, that is all done in association areas, right? Making connections in those areas for those types of learning environments. So we'll go through some of them as we go and talk specifically about some of the association areas. The primary motor cortex we mentioned, this is all of our planned conscious movement, all of our motor commands, right? Our motor commands here meaning our somatic motor commands. So this isn't like churning the stomach, right? This is flexing the bicep. This is skeletal muscle motor commands at our primary motor cortex. And by upper and lower motor neurons, all we're talking about in this concept is where they are. So I'm going to go back to my little